Dr. Fauci, do you still support funding of the NIH funding of the lab in Wuhan?
Senator Paul, with all due respect, you are entirely and completely incorrect that the NIH has not ever and does not now fund gain-of-function research in the Wuhan Institute of Virology.
Do they fund Dr. Barrick?
We do not fund.
Do you fund Dr. Barrick's gain-of-function research?
Dr. Barrett does not doing gain-of-function research, and if it is, it's according to the guidelines, and it is being conducted in North Carolina.
Hi, everyone, and welcome to another edition of Backstory. I'm Dana Lewis. That was Dr. Anthony Fauci and Republican Senator Rand Paul squaring off over the origins of coronavirus. Paul accusing the National Institutes of Health of funding risky Chinese research into bat coronaviruses in Wuhan. The research is called gain of function, which turbocharges viruses to spread to humans. The idea being that scientists can then anticipate the next pandemic outbreak in a laboratory. It's risky science because there have been numerous lab escapes of the viruses. On this backstory, was COVID-19 created in the Wuhan lab? Did it escape? Did the Chinese cover it up? All right, joining me now all the way from Taiwan is Dr. Stephen Quay. Hi, Stephen.
Hi, Dana. How you doing?
Great. Before we launch into the theories of the origins of COVID-19, let's just establish who you are, because it's important, right? Because we're talking about science. So you are part of the Paris group, which, as I understand, is just over a couple of dozen scientists with some of them are mathematicians, they're not scientists. There's a lot of different disciplines in there, but that group was formed because, and what is its raison d'etre?
It's mainly a curiosity around the origin. Everybody has pretty busy day jobs of one form or another, but we've committed to meeting once a month by Zoom for about 4 hours. And we usually have someone in the group, or we have someone outside the group present some scientific aspect of it. And then we have discussions. I mean, we've put out 2 letters publicly to support the WHO and to support further investigations. And that occupies some of our time, the logistics of getting those letters done and the sort.
And you are a doctor of what, and what is really your specialty and your strength?
So I have an MD, PhDs in chemistry. I taught at Stanford Medical School for about 10 years.
I heard of that.
Yeah, it's a pretty good school down there in the mid-peninsula of California. My residency was at the Mass General at Harvard, and I was in a Nobel Prize winner's laboratory at MIT. So during the day, I was learning to be a doctor, and at night, I was doing photochemistry across the river at MIT.
So you're not a virologist. You're not
I'm not a virologist.
Epidemiologist.
I've invented a treatment for a particular influenza using siRNA. So I have 87 patents, and I've invented 7 drugs that are FDA approved, including the gadolinium that's used for MRI contrast. So about 80 million people have used that drug. So I'm a really nerdy scientist. It's probably the core of Steve Quay is. Back in the day, 20, 30 years ago, I would take home before the internet, an inch of scientific papers that I'd Xeroxed. And that was my sitting on the patio with a drink on Sunday afternoon. That's the way I spent my weekend. So fast forward, I now do it on the internet.
Let's talk about the origins of COVID-19, because you are also, I understand, a signature to this letter from, again, about 26 scientists. I don't know if they're the same as the Paris group. I know some of them overlap, which essentially says to the WHO, look, your investigation was flawed from the beginning. The membership that Chinese demanded in terms of the scientists that were on that panel essentially gave the Chinese government a veto. And your findings, you tell me how you want to put it diplomatically, just don't hold water, and we need to start again.
Yes, that's essentially what the letter says. And, there was a second letter that actually tried to offer a blueprint of if you are going to start again, these are the people you need. These are the skill set of the people you involve. These are the steps you should take, et cetera, et cetera. So tried to lay it out pretty clear. So with any sort of impetus, they have the tools they need to do it right a second time.
Where do you line up on the theories? There is one that says maybe it was in bats, and then it jumped to another host, and then it became, it was then transmitted to humans and was able to replicate in humans and grow. The other is a lab escape.
Yeah. So I've written a Bayesian analysis of those two theories. I uploaded it on Zenoda, I think in the end of January. It's been viewed 140,000 times. So just for perspective, I've written about 360 publications, scientific publications on various fields in that. This one is 1 or 2 orders of magnitude better than anything else I've ever written.
So the Bayesian is kind of statistical math formulations on what are the odds, right? And
that is correct.
And I understand that you think through your Bayesian calculations that a lab origin was 99.8% possibility.
Yes, in the legal standard, it's called beyond a reasonable doubt. So Bayesian is a 3-step process. You establish a prior probability based on whatever you can get your hands on. You do an experiment and you see which way it leans. You drop it into Bayes' equation, which is a very simple bit of math. My 8th grade daughter does it, routinely, and then you get a new outcome. So 26 pieces of evidence were run. My starting hypothesis was 98% probability it came from nature. But as you said, I ended up greater than 99% from the laboratory.
Okay, so let's kind of walk through this a little bit. First of all, in terms of hosts, if you go back after an outbreak of a pandemic, you generally find patient 0 and then or patient number 1 or whatever it's called. And then you go back and you find the animal hosts. In SARS 2003, there was an immediate host. That was a cat.
Yeah, it's called the civet cat. It was found 4 months after the first patient.
Then in 2013 with MERS, a camel.
That took 10 months, but you're exactly right. It was the camels.
SARS-CoV-2, we're more than a year into this.
16 months, 80,000 samples of various animals around China. I mean, one of the biases is that you absolutely know China has to be motivated to find it in nature, right? I mean, I think that's an intrinsic either bias or preference. But so after testing 80,000 animals and not finding a single case of it, to me, that's one of the most screaming pieces of evidence that came out of the WHO study that really needs to be addressed.
Chinese would say they just never came from China, that you need to go look in Thailand or somewhere else.
One of the problems with any of this theory is the virus keeps a clock or a calendar or a diary, however you want to describe it. But basically, you can use mutational analysis and phylogenetics to say which virus came before which virus, and therefore put them in chronologic order. That is one of the important things I do in the Bayesian analysis is the genetic patient 0. I don't believe believe it is the actual patient 0 because it's December 20th thereabouts, but was from a PLA hospital. Every patient and every specimen from the seafood market is downstream from that. Every patient.
It's quite close to the Wuhan Institute of Virology, by the way.
Well, they all are, yes. I mean, I actually did another investigation which concludes with what I call the line 2 COVID conduit, which is basically looking at which hospitals where the December patients went, with the assumption that they went to the nearest hospital for where they live, kind of, and then which of the 9 subway lines they were near. All hospitals were along line 2, and that is like a 1 in an 80,000 probability.
Okay, why is it important? Where does line 2 run to and from?
It has some very unique features. So it's the closest station to the Wuhan Institute of Virology. So if a asymptomatic worker came out and took the subway, they would go downstairs. It carries 1 million people a day in normal times, obviously back then before that. So if you imagine it's a back and forth for people from the suburbs going to work, that's 500,000 people. And that's probably coincidentally, but nonetheless, the number of people of infections they said they had in Wuhan was 500,000. It also has the wet market as its closest subway station. And in fact, the WHO said they had a map of the infections inside the wet market, and they're asymmetric. So the west entrance has more infections than the east entrance, and it's the west entrance that is closest to the Line 2 subway. Now it gets really cool for amplification, because the next station from the wet market is the station where the high-speed rail goes. So from that station, you can go to any corner in China that the high-speed rail services in a matter of hours. If you continue to the end of Line 2, you're at the international airport. So you can literally go from the Wuhan Institute of Virology downstairs, never go outside again, and come outdoors in London or Paris or Dubai or Houston, being entirely indoors. So I'm really looking for someone to help me with an analysis, show how much amplification that subway connection probably had. Because it's independent of whether it came from the market or the lab. It satisfies both of those. But carrying 500,000 people a year and having those two major conduits inside of China and into the world, it probably is part of the reason this thing spread so aggressively.
Where do you think COVID-19 began? Some people trace it back to a bat cave, where several miners became sick. I think 3 of them died. And those samples were later taken to the Wuhan lab, where they were doing work on them, trying to understand the bat viruses, and if and indeed it came from a bat at all. It came from a cave.
Yeah. So we know that again, after 16 months and literally thousands of animal testings looking for closest relatives, the closest relative is came from inside the Wuhan Institute of Virology, a bat virus called RETG13. It was collected in 2013 in those same mines where the miners were sick. There were literally hundreds of samples collected, and there are 8 really critical ones that we would love to have the sequences for, because we've seen phylogenetic trees. So Dr. Shi has done presentations, and she's teased us with these other 7 that are very, very close to RETG13, but no one in the world knows, outside of the Wuhan Institute, knows what their sequences are. I think a lot of us suspect that probably a closer relative and the actual precursor to the gain-of-function research that led to SARS-CoV-2 is one of those other 7. My take is RETG is too far. You've got to come up with 1,100 changes to get from RETG to SARS-CoV-2. I can do about 600 with kind of 2 quick things in the lab, but the other 500 are too difficult.
We know that lab at one point was doing gain of function, which is essentially a scientific term where scientists, and it's very controversial. It was banned in the United States at one point, where scientists work with viruses and they try to strengthen them and try and determine when they may, through mutations, jump to humans and thereby head off the next pandemic. And then, there's been a lot of denials by the Chinese that they were doing gain of function, and yet it was published at one point that the bat lady, the woman who ran the lab, essentially was indeed doing gain of function. But did that lead to COVID-19? So do you want to try and string some of that together for me?
Well, there's look at Dr. Shi, Dr. Barrick have worked together. They're 1 and 2, and you can pick which is which. It depends on the day and the year probably, but they're the number 1 and 2 coronavirus scientists in the world, doing synthetic biology, which is gain of function, which is basically taking them apart, putting them back together, mixing pieces from different species of viruses
with human backbone or element.
Well, with they're always a bat virus backbone. So this coronavirus, no matter where it ends up, started at some point in time in bats. And whether the intermediate host is a camel or a civet or something else, in this case, we think it's probably humanized mice. But nonetheless, and then it jumps to humans. But
what's a humanized mouse?
Yeah, it's a little scary. But so you take a mouse embryo and you put human genes into the embryo so that when it's born, it's a mouse, but it has human lungs, respiratory system. So if you want to test what will happen precisely in humans, it's a great model for that. Now, Dr. Barrick developed that model, and his mice physically went from North Carolina to the Wuhan Institute. That's documented again in publications where she thanks him for his mice.
It just sounds like dark arts, quite frankly, but I'm not a scientist.
You know, it is.
And by the way, I mean, I'm not altogether naive either, because I've talked to, I was based in Russia covering biological chemical warfare, the dismantling of the former Soviet Union's incredible programs, and an assistant secretary of defense, not a Republican, a Democrat in that case under the Obama administration, told me that the gain of function research is highly controversial and should be banned, and was banned, and it's high risk. Again, a dark art in essence.
Well, it absolutely is. Again, in my Bayesian analysis, I talk about the fact that it's absolutely published. For 30 years, there has been on average 0.9, so that rounds to one laboratory-acquired infection per year in Asia. So these are SARS-CoV-1, other viruses, one a year. So you're just playing Russian roulette with the 7 billion people on the planet.
There is no good explanation. I'm reading this. This was written by Nicholas Wade, who I interviewed about a week ago. There is no explanation of why a natural epidemic should break out in Wuhan and nowhere else. There is no good explanation of how the virus acquired, and I'm hoping you can explain this, its furin cleavage site, which no other SARS-related beta coronavirus possesses. What does that mean? Yeah.
So it's one of my 5 facts that I talk about that are not in dispute. So there are 5 facts about this event that every scientist agrees to, but they all lead to the conclusion it came from the laboratory. So one of them is this unusual furin site. Okay, so the spike protein is how the virus gets into cells. It's basically like a key, and the lock that the cell has is called ACE2. So it's a protein on the surface of your lung cells, and the spike protein binds to it. And then a second step has to happen where the other part of the spike protein makes a hole in your membrane and injects the RNA. And then it takes over the cell, and then it makes a million of its own copies. So in order for this, so the spike protein is one big protein. So in order to do that physical process of injecting the RNA, it has to actually physically move, and the binding part, you got the binding part to the ACE2, and you got the other part here. This part has to physically move like 100 nanometers, which is a huge distance on a molecular level, to let the RNA go into the cell. So the spike protein has to be clipped. So most beta coronaviruses clip it at a place called S2-1. There's only a couple enzymes that will clip it there, which means there are only a couple kinds of cells that it can go into. So we've known since 1992, 11 different laboratories have done it. Dr. Barrick and Dr. Quay at the Wuhan Institute of Virology. Since 1992, 11 labs have put furin sites in coronaviruses. And 11 out of 11 times, it does one thing. It makes it either more infective, more transmissible, or more lethal, or all of the above. And the reason is it gives a new way of activating that spike protein with an enzyme called furin, which is our enzyme. So basically, the virus is very economical. It doesn't bother having an enzyme to activate itself. It knows with this new furin site that it'll be able to get into a cell.
We've never found furin cleavage sites in natural occurring proteins, or what do you want to call it?
So the virus kingdom is very big. So influenza uses them all the time. Certain other viruses use them all the time. Beta coronaviruses never seem to use them. So we have over 2,000 sequences of beta coronaviruses from nature, and the number of furin sites is 0. Well, it's 1 is SARS-CoV-2. So there's no natural furin site in any beta coronavirus. That's just, I mean, look at 0 out of 2,000. You ask a statistician what the incidence in the population is, and it's under 0.1%.
It tells you what about COVID-19, that furin cleavage site?
So it has it.
And it tells you what about the fact that it has it?
Okay, So if there's, it doesn't.
It tells you it was lab created.
Well, yes. So basically, yes, it's never been found in nature in that class of viruses, and in 11 laboratories since 1992, it's purposely been put including in coronaviruses to make them more infective. So that's one layer of the furin site, which makes it special. I call it the immaculate cleavage site because it literally has to be if it came from nature. The next level is the genetic level. So the furin site is 4 amino acids in a row. Now, I worked with Goldenkron at MIT, who got the Nobel Prize for the genetic code. So there's 3 bases in the code for every amino acid. So it's redundant. So your genetic blueprint that makes proteins has 3 nucleotides, 1 amino acid, 3, 1, 3, 1, 3, 1. So there's 6 different 3-letter words for one of the amino acids in the furin site called arginine. There's 6 different words for the same amino acid. It's redundant. Humans like a particular 3-letter word, CGG. Bat coronaviruses hate CGG. Out of the 64, so if there's 4 nucleotides and 3 letters in a codon, that is 64 different possible codons. So the least popular codon for coronaviruses is CGG. They hate it. They absolutely hate it. And yet here it is. Here are 2 of them together. Now, I've looked at 580,000 codons in beta coronaviruses with the help of a computer, and there is not a single example of the 2 together, CGG, next to each other.
Back to Nicholas Wade's last line, where he says the natural emergence theory battles a bristling array of implausibilities.
Yeah. So I've been working with Nicholas for about 6 months weekly with some of the science behind some of his writing. So yes. So that's absolutely weird. Now, why would the flip side of that is there a laboratory reason why you'd put CGG in? Because if you think about it, if you've got 6 possibilities for the first arginine, 6 for the second, that's 36 combinations, right?
You mean why as a scientist would you put it in?
Yes, exactly. Why as a scientist would you put it in?
Okay, why?
So we know that nature is never going to put it in. In 580,000 codons, it's never appeared there before. So nature hates it. Why in the heck would a scientist do it if there's 36 possibilities?
Answer?
It's very simple. There is an old technique where if you inserted something genetically and you wanted to follow it in your experiments, you would put in what's called a restriction site. So you'd put in a set of amino acids that gets clipped by an enzyme you can buy off the shelf, and then when you run a gel, if the protein if it's still in there and intact, the protein runs very high on a gel, and then if it's been cut accidentally or it's been lost or something, you don't see it. So it's a very common laboratory technique. The Wuhan Institute's been doing it for years, where when you do an insertion, you purposely put in one of the restriction sites so you can follow it in the laboratory. That's the only one of the 36 that has a restriction site like that. So a lot of motivation for putting that in there.
So a lot of the allegations have been that the U.S. National Institute of Health provided funding for gain-of-function experiments in Wuhan. And then you had this very telling Senate Intelligence Committee investigation last week where Republican Senator, Rand Paul then, gets in this, what looked almost like a court examination with Dr. Fauci, who was the head of the National Institute of Health. Will you, in front of this group, categorically say that COVID-19 could not have occurred through serial passage in a laboratory? Fauci, I do not have accounting of what the Chinese may have done. I'm fully in favor of further investigation of what went on in China. However, I'll repeat again, the NIH and the NIAID categorically had not funded gain-of-function research to be conducted in the Wuhan Institute of Virology. Does that ring hollow?
I think it does. I mean, and it may be, in a really lawyerly court-like way, there's a technicality. So what we have is we have the grants that EcoHealth was funded, and we have the subcontracts to Wuhan Institute of Virology inside those grants. So we know exactly what was expected of the WIV inside the grant that EcoHealth got from NIAID, which is Dr. Fauci's work. It's clearly gain of function research. Now, the money didn't go from NIH to Wuhan. It went from NIH to EcoHealth to Wuhan. If that really is what he's relying on, it's disappointing. But I don't have another explanation. Now, he actually also said during that interview that there was no gain of function research being done. And I think on the face of it, on the face of the documents that are available to the public, you don't have to even do a FOIA request. You can just go online in the NIH. You can see they're describing gain of function research in the 5-year grant that funded the Wuhan Institute.
They don't call it gain of function.
They don't call it gain of function, but what they describe is gain of function. So the gain of function is a very broad definition of just increasing the transmissibility, lethality, or infectivity of a virus in the human population, and using humanized mice and in improving in purposely causing testing to test for spillover potential is exactly that. It's again, I will try to pretend to be interested in what they were doing. So the idea was how close to SARS-CoV-1. So we know CoV-1 spilled over. And then we know that there are these viruses that are 60% similar and 70% and 80% and 89%. Literally what they were saying is, what percent does it have to be like SARS-CoV-1 so we know it spills over? And then we can use that number to go out in nature and say, well, here's a virus that's 85, so it's not likely. Here's one that's 92, so it's likely. Giving them the benefit of the doubt, that's probably what they were trying to do. But we know what the outcome is.
Stephen, just a couple of quick questions as well before I let you go. The data that was online, some of it from the Wuhan lab, some of it that you required a password, I understand, all disappears in September. It's taken down in September. So October, November, some 3 months before the official outbreak, although a lot of people think that they already had cases well into November. What do you make of that, and how important is that data?
Critically important. 3 a.m. September 19th, a server that was available for the world for most of it, and it also did have a password protected part, but Steve Quay could have gotten on it the day before, but it's taken offline. Now they slipped up and they said, well, it's taken offline because of hackers around the COVID infection, and then they realized, oh my gosh, this was in September. So I'm not sure they're still saying that.
That's a pretty big slip up.
Well, it's out there. That's how it is with cameras. Anyway, but and it's not available to this day. So I asked Peter Daszak in a public forum here maybe 2 months ago, where if he would provide access to those files, and he literally said it's on the record there that he knows what's in them. He's looked at them, and there's nothing there of interest. And that's the basis for not providing them.
Well, then show them to the world, because right now that lab is under. Look, I think you tell me if this is an overstatement, but it seems to me that there is a consensus amongst the scientific community now that at the very least there are huge, gaping gaps in the Chinese official story on that lab. So if you want your lab, if you really want to support your suggestion to the world that it didn't originate in the lab, then make all the data available. That's the best thing that they could do right now to the WHO or whoever.
Agreed. Whether they would do that or not is a political question.
Right. Okay. So but that data does still exist, we think.
Oh, I hope so.
Are we ever going to get to the bottom of this?
Well, see, that's an interesting question, because with my analysis, all of my evidence is circumstantial, but I remind people that, there are many sort of murder trials in which all of the evidence is circumstantial, and yet we quite routinely can determine the guilt of someone with a proper process and adjudication and getting to beyond a reasonable doubt. So despite it being circumstantial, I believe it's strong enough at this point in time to conclude, again, beyond a reasonable doubt, that it came from the laboratory. The key thing to remember with these coronavirus pandemics is there's 3 components. there's the intermediate, there's the host, the animal host, the humans, and the virus. This is the only one in which it's a singularity. That is, there seems to be only one animal because we can't find any in nature. So there's one animal. All the human cases go back through one patient at the PLA hospital in Wuhan. So when President Trump got it, you can follow the mutations to his virus back to the PLA hospital. So there's a singularity there. And the sequences inside the virus. The patient. There's absolutely no patients before December who had any hint of this infection. So it is a true singularity.
Which I understand is quite interesting.
One animal. One patient, one virus, one animal. And
because normally it takes a virus time to become more and more transmissible. There should be a trail of patients.
Oh, yeah, exactly. And again, so in my Bayesian analysis, I looked at stored specimens in Wuhan and China. I had about 2,000 of them in the public record, none of which had SARS-CoV-2. The WHO has actually updated that. So there are 10,000 specimens that they reported on in their report with zero seroconversion before the first date. So nobody out of 10,000 in archive specimens had SARS-CoV-2. What is the MERS or SARS-CoV-1 history there? 1 to 4%. So that means 100 to 400 samples there should be evidence of SARS-CoV-2 in 100 to 400 specimens out of 10,000. There was zero.
The reason why we just spent 30 minutes talking about this and why you're spending part of your career studying this and why so many other scientists are looking at it is because in order to understand the next pandemic, you have to you need to trace back the origins of this one. Can I ask you something that occurs to me, and you can tell me whether it's ridiculous or not? But in trying to fight this pandemic and come to terms with what COVID-19 is it important that we understand it was created in a lab? Does it make it a very different pandemic and a very different fight?
It has one. It seems to me it has one aspect of the design of a virus that causes a pandemic that I think is interesting and worth talking about. So the downside is that it is clearly pre-adapted for humans. So when SARS-CoV-1 first jumped into humans, it had only 17% of all the mutation changes it needed to cause that initial epidemic, 17%. This one has 99.6% at the get-go. So the downside is it was very pre-adapted, so it spread very quickly, very widely, and that was the first wave of it. There actually is an upside to that, which is it has very little genetic runway, very little movement to improve. So in the spike protein, there are only five positions that it could mutate at to get better at from the initial case. It turns out what we called the UK strain, that the British strain that was started in the fall and spread, that was one of the five. So and now
what about the Indian? What about the India variant?
That's another one. So it's used up 2 of the 5 possible improvements in the spike protein in terms of binding. So that's somehow to me encouraging that this thing, that we've seen the worst of it, and these variants are not going to be able to have much impact. I did another analysis, and there's 8 epitopes. So the human immune system sees 8 separate things on the spike protein when it sees this virus. So it makes 8 different kinds of antibodies. And at this point in time, the Indian strain has only gotten rid of 2 of those 8. So we still have 75% of the epitopes are the same. When I was teaching at Stanford, 75 was a C, but in immunology, that's a pass-fail, and that's a strong pass. You probably have to get well below 50% to have an issue there.
Does that give you a roadmap on where we will be in a year or 3 from now with this virus?
I said a year ago that unfortunately 7 billion people have to either get the virus or get the vaccine. And I'm afraid I haven't changed that tune yet.
And we're years away from that.
That is unfortunate. That's the analysis, Dana.
Stephen, last word to you, and I thank you for your time on all of this, just wrapping it together.
Well, I think that my belief is that it's a gain-of-function research project. My belief is that there is a way forward for gain-of-function research to be done on a different structure. I'm going to be proposing that publicly in the next few months when I put together a consortium around how that should be conducted. But I think absent that, we're just going to do this again with another virus in another decade or another few years.
Or just ban gain of function altogether?
We could. That would be the easiest. I'm willing to find a middle road if that is possible.
Dr. Stephen Quay from Taiwan. Stephen, great to talk to you. Thank you so much for taking all that science and trying to make it as digestible as possible, and I appreciate it very much.
Thank you, Dana, for focusing on this. It's an important issue.
So why was there a scientific split on using the term gain of function in the Wuhan lab? Why do we hear Dr. Fauci say there was no gain of function? Well, I'm no scientist, but Dr. Richard Ebright, a professor of chemistry and chemical biology at Rutgers University and a biosafety expert, contested Dr. Anthony Fauci's testimony before the committee, saying that Fauci's claim, made during the exchange with Rand Paul, that the National Institutes of Health has not ever and does not now fund gain-of-function research in the Wuhan Institute of Virology is demonstrably false, according to Ebright. At least some of the NIH-funded research conducted in Wuhan equivocally qualifies as gain-of-function, Ebright was quoted as saying. Ebright claims that the work being conducted at the Wuhan Institute using U.S. funds epitomizes gain-of-function research and is the exact kind of research that led the Obama administration to conclude that gain-of-function was too dangerous to continue domestically. And it was in fact banned in the US in 2014 following a series of lab accidents and after a petition signed by 300 scientists demanded a moratorium on gain-of-function. The ban was later lifted after a review by a secret government panel, and gain-of-function experiments were allowed to go forward. The Obama administration's ban was lifted in 2017 under then-President Trump. That's our backstory on the origins of COVID-19. Subscribe to our newsletter if you like, danalewis.substack.com, and please share this podcast so that others can listen. I'm Dana Lewis. Thanks for listening, and I'll talk to you again soon.